Good morning. We cannot survive without eating or even drinking. While are eating, some juices or some enzymes are put inside certain system which is responsible for digesting your food. This is done inside special organs inside your digestive system. Before swallowing and before digestion, the masses of the food inside your mouth are found in the form of big pieces or big parts. Through a long journey, these big pieces or parts are transformed by the time into smaller particles of food until we can benefit from it. What happens inside this long journey? Some juices or some enzymes are put inside the digestive system, changing the large molecules of the food into very small one to allow the body to benefit from. Here we have some juices inside our bodies which are acting on different kinds of the food. This is done by enzymes. The enzymes are biological catalysts. They are made of proteins. These enzymes are responsible to change the large molecules of the food into very small ones so we can benefit from. We cannot depend on the large molecules of the food as they are inside our mouth, but they must change into smaller particles to allow the small molecules to transfer from the small intestine to the bloodstream to let the body benefit from. So the enzymes are protein, biological substance, or a catalyst. You know that the catalyst is a chemical substance which activates and speeds up the chemical reaction, but it doesn't change its quantity and property. Once again, the enzymes are protein, biological substance which activates and speeds up the chemical reaction without being used up or even without changing in any quantity and property. So, the enzymes are acting on different kinds of the food. The food here is called a substrate. This word means different kinds of food. You know that the body needs different kinds of the food groups like proteins, sugar, carbohydrates, fats, and so on. Each one of them we can call them a substrate. The enzymes which are acting on different substrates are not the same, because the enzyme which is acting on the starch food or the starch substrate is different from the enzyme which is acting on the protein substrate. So the enzymes are specific. Specific, it means that each enzyme is acting on a certain substrate. The enzyme which is acting on the starch food is not capable to digest the same substrate of another group of food like proteins. Here we have some examples. For instance, you know that the starch food from the previous discussion are supplying the body with energy, carbohydrates including sugar and starch. Even the fats supply the body with energy. The enzyme which is acting on the starch food is completely different from the enzyme which is acting on the fats. Try to have an idea about the difference between the action of the enzymes on different substrates. You know that iodine solution can be used to detect or to test for the starch substrate. If you put some starch in a test tube containing water and shake it very hard to form a starch solution, then on adding drops of iodine solution inside the test tube that contains starch solution, what will happen? You can observe that the color of the starch solution changes from transparent to dark blue. Let's try another experiment. If you prepare a starch solution, starch with water, and shake both of them together strongly. Then if you add droplets of saliva from your mouth and shake it once again, then add drops of iodine solution. This time the color of iodine solution will not change into dark blue. This is because saliva inside your mouth contains certain enzyme called amylase. So before adding saliva to the iodine solution and starch solution, the iodine solution changes into dark blue, but after adding saliva solution to the starch solution, the color doesn't change. This is because the amylase enzyme, which is existing in the saliva in the mouth, secreted by the salivary glands, changing the molecules of the starch into simple sugar. So the action of the amylase breaking down the starch into simple sugar molecules or simple glucose. That's why the color of the iodine doesn't change. Here we have another example of the enzymes, protease. Protease is an enzyme which splits the protein molecules into amino acids. Another example of an enzyme, the lipase enzyme. Lipase is active on lipids or fats. It breaks down the fats into glycerol and fatty acids. This is a completed molecule of fat. By the action of the lipase, it will be broken down into glycerol and fatty acids. Glycerol is separated from the fatty acids. Don't ever forget that starch is digested in two regions. In the mouth, by the action of the saliva, and in the small intestine, by the action of the amylase secreted by the pancreas. We are going to explain it more and more and more in the next lesson. So starch can be digested in two different regions inside your body, in the mouth and in the small intestine. Proteins. The protein substrates can be digested in two different organs inside your body, in the stomach and in the small intestine. Finally, the fats. Fats substrates can be digested only in the small intestine. You can recognize that in the small intestine, food is completely digested and absorbed. After food is completely digested, it will be ready to be absorbed to the bloodstream. That's why the action of the enzymes on different substrates is to change the large molecules of the food into very small one, so that they can transfer from the walls of the small intestine to the bloodstream. Why? To let the body benefit from. The enzymes can work only on the molecules. These molecules are found on the surface of the food substrates. But in some types of the food, like fats for instance, the molecules of the fats are concentrated inside the substrate itself in the middle. So we have to chew the food very well. You are going to make chewing very well before swallowing. That facilitates and makes the digestion process is going easier and faster. And that helps the actions of the enzymes to do their functions very well. You know, of course, that swallowing the food in big parts and swallowing the food in very small parts, which one of them is preferred? Of course, by chewing the food very well, you are going to change the large molecules of the food or the large molecules of the substrates into very small ones. That helps the action of the enzyme to act and to function very well. Finally, by chewing the food by the teeth very well inside the mouth before swallowing, that increases the surface area, which allows the action of the enzymes to function well. I have just mentioned before that the substrate of fats, or the fat substrate, its molecules of this substrate are concentrated inside itself. So in order to act as the enzyme like the lipase, in order to work properly on the fats, it should be divided into smaller particles. This is done by, yes, bile juice or bile salts. Bile is not an enzyme. Bile is stored inside the gallbladder. I'm going to explain in the next lesson, which is located inside the liver. Bile juice is produced inside the liver and stored inside the gallbladder. The bile salts are acting on the fat substrate, dividing them into smaller molecules of the fats, but we cannot consider the bile salts as enzymes. It just increases the surface area of the fats to facilitate its action. Any enzyme is affected by two important factors, temperature and pH. The enzymes are working properly between the degrees from 0 to 40 centigrade. The action and the activity of the enzyme is working properly between this range. But what happens if the temperature is becoming above 45 or 50? The action of the enzyme decreases and denatures. It means it doesn't work properly at all. This figure represents the graph and the range of the activity of the enzyme. Look here, when it starts from the 0 temperature, when it increases to 10, the action of the enzyme increases, the activity of the enzyme on the substrate increases until it reaches the optimum at 40 centigrade. When the temperature increases above 40 centigrade, the action of the enzyme, the kinetic energy of the enzyme and the substrate starts to decrease until it denatures above 50 centigrade. So above 50 centigrade, the enzyme loses its activity and denature. This is concerning to the temperature as a factor affecting on the action of the enzyme. Here we have another important factor, the pH. You note that the range, this range from 1 to 12 is indicating the medium in which the enzyme is working inside. From 1 to 7, we can call this area is acidic medium. Above 7 to 12, a basic medium or an alkaline medium. What about this range exactly, 7, neutral medium? What does it mean? It means that some enzymes are working in an acidic medium, but some enzymes are working in a base medium. Some enzymes are neutral. Here we have 3 examples of the enzymes: pepsin enzyme, amylase enzyme, and arginase enzyme. If you are looking at the optimum point of the enzyme action of the 3 enzymes, number 1, pepsin enzyme, its maximum or optimum activity of this enzyme will be at 2, because this number is below 7. So its action is working properly in an acidic medium because it's below 7. This one, the second one, amylase, the amylase, its optimum point of activity is at 7. So its optimum activity range is neutral. The last one, the arginase enzyme, its optimum activity range will be at 9 or even 10 here. So its medium of activity or action is a base.